After gastrulation, we begin organogenesis. So we're starting to form the rudimentary beginnings of the organs. And the mesoderm is going to form into the notochord, and then the ectoderm outside of that is going to form the neural plate, which begins curving until it forms a groove, and then those lips are going to come together to form a tube called the neural tube. That eventually will become your brain and spinal cord. And then around the notochord, you form these somites, which like what you see in a cephalochordate, are going to form segmented muscles. So here you can see this notochord and the neural tube around it, again, which started off as just a groove, and then the groove, as it invaginated, came together to form a tube. And you can see these somites forming around the outside of that. You can see how they're segmented right there later in organogenesis. And there are a lot of similarities in terms of the mechanisms of organogenesis between invertebrates and vertebrates, but there are big differences in the ultimate body plan that you see, particularly if you look at the neural tube as an example. So a lot of the invertebrates that we've talked about in the last couple of weeks had ventral nerve cords, whereas vertebrates are chordates, which we know have dorsal hollow nerve cords. And as these cells sort of move and reshape during morphogenesis, during gastrulation and organogenesis, you might wonder, well, how do these cells do that? And one of the main forces behind this is reorganization of the cytoskeleton within the cell to change its shape. For instance, if you look at these layers of cells, the cells right here have extensions of actin filaments that grow larger and larger and larger, specifically in the middle and less so on the side, and that changes the relationship of these cells to these others, and it causes them to sort of grow inward like this, and that forms this neural plate and ultimately groove, the neural groove, and then ultimately it forms that tube. As you see this groove, these lips come together and form the neural tube, which will ultimately be the central nervous system. This all has to do with growth, asymmetric growth of cytoskeletal elements within the cell. Additionally, some cells are intended to die or be engulfed by neighboring cells at certain parts of development. So, for instance, if you have extra neurons that aren't supposed to be there, they'll be removed by apoptosis. When we form our limbs, they are held together sort of by webbing tissue as our digits form, and that webbing tissue is destroyed by apoptosis. So this is programmed cell death. These cells are programmed themselves to die at a certain part of your development. As you are undergoing development, your cells will undergo determination. And determination is the process by which they are sort of committed on a particular fate of development. And then differentiation is the process by which they specialize themselves in structure and function. So, analogy your book uses, determination is like declaring a major in college, right? That sets you on that path, and differentiation is like taking the classes along the way in college to get your specialized knowledge. And you have a lot of different kinds of specialized cells and tissues, but all of your cells have the same genome, the same 46 chromosomes with the same sets of genes on them. So it's not a matter of having different genes, it's a matter of expressing different sets of genes in different kinds of cells, turning on certain genes and turning off other genes. And biologists can create fate maps, which are diagrams that show essentially which structures and organs are going to arise from which parts of an embryo. And there are a lot of classic studies looking at frogs showing the cell lineage from germ layers all the way back to the blastula cells. You can trace them from the blastula cells through which germ layers they're going to become. So here you can inject with dye cells within the 64-cell embryo, and then you can see later on which cells these turn into. And there's consistency with this if you do these studies, right? So these cells then you would see here. And so this is an example of one of these fate maps. And C. elegans is an excellent example of fate mapping because it forms eventually exactly 959 cells in an adult. So it's very small. But developmental biologists were able to fate map every single one of these 959 cells ultimately from the blastula and the zygote. So you start with the zygote and the first cell division, and again, scientists have fate mapped cells to go to each system, right, including the intestines. It's remarkable. And the other nice thing about C. elegans, this is one of the first organisms that had its genome sequenced. And so now scientists are looking at the genes that affect this differentiation. In bilaterally symmetrical animals, which is most animals, you begin forming those axes early on in development. So if you are bilaterally symmetrical, your left-right axis is largely symmetrical, right? My left side is essentially the same as my right side. But my dorsal side and ventral side are not the same. And my anterior side and posterior side, which I won't put on the camera, are also asymmetrical from one another. And these axes are formed, again, early, like anterior posterior is formed during oogenesis. And the animal-vegetal asymmetry, those two different ends of the egg, which show different patterns of mitosis, indicate where the anterior-posterior axis is going to form. The dorsal-ventral axis is formed still pretty early, but not until after fertilization, rather than just when you're an egg, pre-fertilization. Determination of cells happens pretty early on, but not at the very beginning, at least in deuterostomes, because we have indeterminate cleavage. So in a frog embryo, the first two blastomeres, so after that first cleavage, both of them are totipotent. Totipotent means you can become any kind of cell type in the body. And so if those first two blastomeres break apart, you can form identical twins. In mammals, it goes even further than that. We remain totipotent through the 8-cell stage, which is longer than other organisms. And again, this allows, if those cells break apart, for both of those clumps of cells to each form an entire organism. And that's why you can have monozygotic or identical twins, which come from a single sperm and a single egg forming a single zygote, hence monozygotic. That zygote undergoing cleavage, breaking apart, and forming eventually two genetically identical organisms. So identical twins are clones of one another. And this can't happen unless you have cells that are totipotent through at least the first cleavage or first couple of cleavages.